Welcome back to Intro to HACCP. Today we'll be talking about food safety hazards as we get ready to do a hazard analysis. In the previous lesson, we discussed the purpose and importance of preliminary tasks in developing a HACCP plan. We talked about 5 preliminary tasks. Can you remember what they are? Take a minute to see if you can remember. The 5 preliminary tasks of developing a HACCP plan that we discussed in the last lesson were assembling a HACCP team, describing the food and its distribution, describing the intended use and the consumers of the food, constructing a flow diagram, and verifying this flow diagram. Now we're getting closer to the first of the 7 HACCP principles, hazard analysis. Before we discuss hazard analysis, we need to stop and address the types of food hazards that exist in foods and food processing. After studying this lesson, you should be able to differentiate between the major categories of food safety hazards, identify potential hazards in a process and determine if they are likely to occur, and outline basic control measures for food safety hazards that are likely to be controlled by critical control points. Let us begin this lesson by defining food safety hazards as any biological, chemical, or physical agent that is likely to cause illness or injury in the absence of its control. These types of agents, biological, chemical, and physical, serve as the main categories used to differentiate hazards in a HACCP plan. Let's discuss them one by one. What comes to your mind when you think of biological hazards? You probably first think of bacteria such as Salmonella or E. coli that are often in the news associated with some sort of foodborne illness outbreak. Do you know what term is used to categorize disease-causing microorganisms such as these? The term is pathogens. Biological hazards can exist in 4 forms, the most common being bacteria. Can you name the other 3 forms of biological hazards? We'll be talking about them one by one. Pathogenic bacteria are a major concern because they are known to cause 3 types of foodborne illnesses. Eating foods contaminated with living pathogenic microorganisms causes foodborne infections, in which the microorganisms colonize in the host's intestines. Examples of microorganisms capable of this type of illness include Salmonella species and Listeria monocytogenes. The other 2 types of foodborne illnesses involve toxins produced by bacteria. Certain bacteria produce toxins that are harmful when ingested. Intoxications occur when foods containing toxins produced by bacteria or mold are eaten. The microorganism itself does not need to be present to cause illness, just the toxin. Examples of bacteria that cause food intoxications include Staphylococcus aureus and Clostridium botulinum. Toxin-mediated infections are similar to intoxications in the sense that toxins cause illness. However, they are different in the sense that the microorganism produces the toxin while inside the human intestines. These infections are also similar to foodborne infections because the bacteria itself can cause illness by colonizing inside the intestines. Clostridium perfringens and E. coli 0157H7 are examples of microorganisms that cause toxin-mediated infections. The other 3 types of hazards of biological origin are viruses, parasites, and molds. Let's talk about viruses. Viruses have a wide range of health effects. Some viruses come from the meat of diseased animals. Many viruses that are major concerns in the food industry do not come from the food, but from contaminated waters, such as sewage contamination, or ill food handlers instead. Parasites cover a broad range of the animal kingdom. Many have complicated life cycles that can allow them to infect humans. Primarily, the parasites are roundworms, single-celled protozoa, tapeworms, and flukes. One group of parasites that is important in the meat industry is trichinae. These are tiny roundworms found in the muscle of infected animals, usually pigs, which cause trichinosis, a disease characterized by intestinal disorders, fever, muscular swelling, pain, and insomnia. Infection by parasites can come from eating raw or undercooked meats and fish. Consumption of raw produce can be traced to infections of protozoans also. The fourth biological source of hazards is mold. Accounting for hazards presented by molds in a HACCP plan can be confusing, since molds produce toxins which are often considered as chemical hazards and not biological hazards. Mold toxins are called mycotoxins, and they can cause acute poisoning or chronic health effects such as cancer over a period of time. In apples, the growth of Penicillium expansum, a mold responsible for blue mold rot, is responsible for producing the toxin patulin. Patulin is a mycotoxin produced by fungi belonging to several genera, including Penicillium, Aspergillus, and Bysoclamus. Patulin can occur in many molding fruits, grains, and other foods, but the major source of patulin contamination is from apples with brown rot, so it is found in apple cider and apple juice. Patulin is stable in an acidic environment and is not destroyed during thermal processing. The growth of the fungus Aspergillus flavus on grains consumed by livestock can cause aflatoxin residues in meat and milk products. Recent work has also shown that spices added to meat products can be a significant source of aflatoxin if the spices had been contaminated by mold. Let's move on to chemical hazards. Chemical hazards can come from many sources. Some chemical hazards are the result of unintentional adulteration of the food with toxic chemicals, while some may be present in meat or milk products as therapeutic drug residues. Normally, pesticides intended for use on food crops are not considered hazardous when properly applied. Pesticides that are misused can become chemical hazards in foods, especially when they are not intended for use on food crops or have been misapplied to foods or feeds. Processing chemicals, such as cleaners and sanitizers, are not usually considered toxic compounds when used as intended. Even when they adulterate products, they are not usually considered toxic. Some boiler treatment compounds may leave toxic residues in steam if they are not intended for the production of culinary steam. Therapeutic drug residues may be hazardous when misused as well. Government agencies monitor the presence of certain drug residues, especially antibiotics, in meat and milk. Another category of chemical hazards is that of naturally occurring hazards. Some foods, such as polar bear liver, may have toxic levels of vitamin A. Potatoes can produce a natural alkaloid, solanine, which can cause illness or death. Marine toxins are another source of natural occurring hazards. Examples of these include scrombrotoxic fish poisoning, ciguatera poisoning, paralytic shellfish poisoning, neurotoxic shellfish poisoning, and amnesic shellfish poisoning. Some foods may cause allergic or sensitivity reactions in susceptible persons. Usually these persons are aware of their susceptibility and are careful to avoid these foods and ingredients. A hazard exists when undeclared allergens are present in a food product, either from accidental adulteration or from failure to list an ingredient that is an allergen on a food label. At last, we're on to physical hazards. Physical hazards result in choking or injury. These hazards may come from materials naturally present in the food, such as pits and bone fragments, or they may come with the food, such as broken injection needles, bullets, and buckshot. Other materials may come from handling or processing equipment, things such as staples, twist ties, pop-up timers, broken glass shards, bolts, nuts, and saw blade pieces. Now let's switch gears and talk about how to control all of these hazards, starting with microbial hazards. First, I want to share 2 resources that will be invaluable to you. These are required reading assignments. The first one is the Bad Bug Book provided by the FDA that describes all of the pathogens found in food and how to control them. The other one is also by the FDA called Factors That Influence Microbial Growth in a guidance document. You should find the links to both of these websites on the Moodle site. Raw products produced under good agricultural practices are most likely to exhibit a lower concentration of pathogens due to the care taken in production. Once the raw materials are transported to the processing facility, the microbial concerns generally fall into the following categories: pathogens present on the product from the field or from transportation, growth of pathogens on the product due to improper handling and storage conditions, contamination from handling, equipment, water, and other plant sources, destruction of competing microorganisms from treatments or under processing, growth of pathogens in the absence of those competing organisms, and toxins produced by the growth of toxin-forming microorganisms. The most obvious control measure available to the processor is to minimize contamination of the raw materials or the product with pathogens. In the plant, this is accomplished by using clean equipment and utensils. Sanitizing can reduce the pathogen loads on processing surfaces. Raw ingredients and water must be of good quality when performing sanitation. Growth of pathogens can be inhibited by controlling the conditions of storage of food products and ingredients. The FDA's Bad Bug Book lists the growth temperature range of some common pathogens. You will note that most of the pathogens will not grow appreciably at refrigerated temperatures below 5 degrees Celsius or 41 degrees Fahrenheit. Those that do grow are considered psychrotropic pathogens, and they do not grow well in the presence of other competing microorganisms. The upper limit of pathogen growth is about 45 degrees Celsius, or 113 degrees Fahrenheit, except in the case of certain spore-forming pathogens, which can grow up to 52 degrees Celsius, 126 degrees Fahrenheit. Generally, food scientists speak of a temperature danger zone, which reaches from 41 degrees Fahrenheit to 135 degrees Fahrenheit. This was previously 41 degrees Fahrenheit to 140 degrees Fahrenheit, but it was recently changed. Food which is likely to support the rapid growth of pathogens should not be held within this range for more than a short time, usually defined as more than 4 hours. Thus, rapid cooling of cooked and processed foods through this zone is important. Temperature may be the most important factor affecting microbial growth. Since environmental temperature can control the types of organisms able to grow and survive on a food product, we classify organisms by their temperature requirements. Organisms that grow well in low temperatures are called psychrophiles. Organisms that grow well in ambient temperatures are called mesophiles. And organisms that grow well in very hot environments are termed thermophiles. Classification is based upon optimal growth temperature and the temperature range over which bacteria grow, not just survive. For example, mesophilic Bacillus and Clostridium species can survive very high temperatures because of their spore forms, but are still classified as mesophiles since they can only grow in the mesophilic temperature range. Like we said, psychrotrophs are those organisms which have an optimum growth temperature below 25 degrees Celsius, or room temperature. However, many mesophilic organisms can also grow in this range. We are particularly concerned with those pathogens that grow at refrigerated temperatures, because many foods are held in the refrigerator. Thermoduric organisms are organisms that can survive the pasteurization temperatures. These are obviously of special concern as well, since many foods are pasteurized. Most pathogens are killed by temperature treatments at 140 degrees Fahrenheit for 30 minutes. A temperature of 161 degrees Fahrenheit for 15 seconds has a similar lethal effect on pathogens. Some viruses may require higher temperatures or longer time for destruction. Some pathogens produce spores, which are a form of the organism which is resistant to many adverse conditions, such as heat, acid, and desiccation, or drying. Once the vegetative cell, or the rapidly growing form of the organism, is destroyed, the spore may still be viable and will form more vegetative cells when conditions are suitable. Spores require extreme processing conditions to destroy them. Since I've been talking about spores, I have not defined spores, so let's talk about spores for a while since they're so important in terms of temperature. Some bacteria can produce a protective spore structure. A spore structure allows a cell to withstand environmental stresses such as cooking, freezing, high salt, drying, or high acidity. Generally, bacterial spores are not harmful if ingested. However, if conditions of the food are changed that permit the spore to turn into a vegetative cell, the vegetative cell can grow in the food and cause illness if eaten. Spore-forming bacteria are generally found in ingredients that are grown near the soil, like vegetables and spices. When environmental conditions improve, such as the addition of dried spices to a soup, spores can become vegetative cells. For an example of talking about spores in foods, I want to talk about one of my favorite meals, chili. When making chili, all the ingredients, such as beans, meat, spices, tomato paste, and cheese, are mixed together and cooked to a rapid boil. At that rapid boil, which is somewhere around 212 degrees Fahrenheit, all the vegetative cells are destroyed, but the spores may survive. If I were to make some chili and have some chili with my friends, eat it, and put the rest of the chili in the refrigerator, it might take 8 hours to cool that chili from, say, 135 degrees Fahrenheit to below 41 degrees Fahrenheit. If given enough time at the right temperature during this cooling process in that temperature danger zone, spore-forming bacteria that survive the cooking process may change into vegetative cells and grow. This is called outgrowth of spores. So, to keep spore-forming bacteria from changing to the dangerous vegetative state, or to prevent outgrowth, it is critical that hot foods be maintained at 135 degrees Fahrenheit or above, and cold foods be maintained at less than 41 degrees Fahrenheit. This is where the temperature danger zone concept comes from. Important spore-forming pathogens include Bacillus cereus, Clostridium perfringens, and Clostridium botulinum. Now let's talk about pH, which can be a factor in inhibiting the outgrowth of spores in a food product. Also, many microorganisms produce acid conditions which limit the growth of the vegetative forms of certain bacteria. In reading the required readings that I mentioned earlier, the FDA guidance document and the Bad Bug Book, you will note that the growth of common pathogens is inhibited below pH 4.4. At pH 4.6 or below, Clostridium botulinum spores will not grow and produce toxins. The pH is an easily measured factor and is useful to determine whether a product is likely to support the growth of pathogens. It is important to note that even though they cannot grow, some pathogens may survive pH conditions as low as 3.5 for days and still can cause foodborne illness. So it's important to consider time at pH. The pH of a bacteria's environment affects both its rate of growth and its viability. Bacteria can grow within certain pH ranges that are different for different species. However, most bacteria grow best at pHs near neutral, or somewhere between pH 6.6 and 7.5. This is the pH in most meats, by the way. As the pH of the environment approaches either the lower or upper limit, bacterial growth slows and eventually the bacteria will die. Some foods, such as fruits, have a pH of 3 to 4.5, and so they have a naturally low pH that resists bacterial growth and spoilage. Bacterial growth generally occurs between pHs 4.5 and 9, but molds grow well between pH 1.5 and 9, and yeasts grow well between pH 2 and 8.5. So it's important to consider the growth of yeast and especially molds, since molds can produce toxins. Moving on to water activity. The role of water in a food is important from a microbiological standpoint. The availability of water to support microbial growth is measured in terms of water activity. Water activity is a critical factor that can affect the safety and shelf life of foods. It measures the free or unbound water present in a food on a scale of 0 to 1.0. Water activity is a better indicator than total moisture content in predicting stability and microbial growth in a food product. Most bacteria will not grow at water activities below 0.91, and most molds cease to grow at water activities below 0.8. By measuring the water activity of a food, it is possible to predict which microorganisms will and will not be potential sources of spoilage. Water activities below 0.85 in a product will not support the growth of pathogenic bacteria. One pathogen that grows at water activities down to 0.87, however, is Staphylococcus aureus. This organism can produce a heat-stable toxin. The last way to control microbial hazards in foods is by using preservatives. Most common food industry preservatives are designed to inhibit the growth of yeasts and molds. They work best at optimum pHs, and when combined, their effect can be synergistic. This is the case when potassium sorbate and sodium benzoate are added together, for example, when controlling the growth of fungi. Now let's return to chemical hazards. We want to be very careful with all the chemicals used in the processing environment, as well as the food itself. So, suppliers' guarantees and certificates of analyses are helpful to minimize the adulteration of chemical hazards on raw materials. The best method to prevent the unintended introduction of chemical hazards in the processing plant is to isolate and clearly label those compounds which may be of concern. In the case of ingredients which may cause allergic or sensitivity reactions, it is important that there be an effective cleaning process after their use on shared equipment. Usually plants will use a production matrix to keep track of sanitation and cleaning schedules. Quick tests for assessing the presence of residues may be employed to test the effectiveness of cleanups. Finally, let's return to physical hazards. As was the case with chemical hazards, requiring suppliers of raw ingredients to provide suppliers' guarantees and certificates of analyses is a good way to show that there are no physical hazards in the food. Proper maintenance of equipment and regular inspection of choppers, grinders, and saw blades also can be used to minimize the potential of metal or plastic fragments which may find their way into the food. When glass is used, there must be an effective glass control program to keep glass out of the product. Also, the effective use of screens, filters, and separators can remove certain physical hazards. Magnets can prevent the contamination from many metal objects. A properly functioning metal detector can also help to minimize the presence of metal in the finished product. Last but not least, good manufacturing practices can be used to control the risk of physical hazards. We don't want our personnel working in the facility to have zippers, rings, or earrings, or even bracelets in the presence of the food processing environment. And so that's why we ask visitors and plant personnel, or the line workers, to take off their jewelry before entering the facility. I want to leave you with a question for thought, and there's not necessarily a right or wrong answer. This is just an interesting topic that comes up just about always in developing a HACCP plan. The question is, should metal detectors be listed as a physical hazard in a HACCP plan? Metal fragments are often listed as potential physical hazards on a hazard analysis. These fragments can often come from several sources. Strangely, other fragments, such as glass, plastic, wood, stones, and bones, are often ignored in the same hazard analysis. while the metal is designated to be likely to occur. This makes you wonder why these other physical hazards are not listed. Should they be listed, or should the metal hazard not be listed? That concludes this lesson. So in summary, we talked about the 3 food safety hazards, being biological hazards, chemical hazards, and physical hazards. We talked about the 4 origins of biological hazards, being pathogens, parasites, viruses, and molds. As far as pathogen-related illnesses, we talked about infections, intoxications, and toxin-mediated infections. You should be able to differentiate these 3 terms. We talked about the numerous methods to control microbial hazards, those being sanitation, temperature, pH, water activity, and the use of preservatives. And finally, we talked about how to control chemical hazards and physical hazards. So now you should be ready to take on our next lesson, which is on hazard analysis.